Until now we have been talking about materials, which is one of the three different aspects of tissue engineering, right. We will now move on to the next aspect, which is cells. Any tissue engineering application, you want cells along with the scaffolds. Many a times cells are also used by themselves without a scaffold, just for regeneration of tissues. It is important for us to know what types of cells are being used, where we can get them, and how we can harvest them, and whether we can culture them to get more number. Because we might not be able to get enough numbers just by isolation from host tissue. We might have to culture them. It is important for us to understand all these concepts. We will first start with the source of cells. Cells are basically of three different types. We have already discussed this earlier. You have autologous cells, which are cells from the same individual, allogenic cells, which are cells from another individual of the same species, and xenogenic is cells from another species altogether. There are different sources for these types of cells. You could either have differentiated cells of the tissue type, or you could use stem cells and fetal cells or embryonic cells, or you could use cells which are of a different type, in the sense that you might be trying to you might be trying to engineer bone, but you would probably have osteoblasts, which is the type of cell present in bone, along with endothelial cells, which is not specific for that bone for the tissue type, but it has its own role in tissue engineering, right. You could use cells of this kind of types. There are also studies where people have shown that one type of cell can be converted to another type of cell, even if it is a somatic cell, means without actually taking them to the pluripotent state. You could just trans differentiate them from one to another type. Okay, there are different things which people have worked on. We will try to understand some of the fundamentals and take it from there. Autologous, allogenic and xenogenic have their own advantages and disadvantages. With respect to autologous cells, the biggest advantage is there is no risk of disease transmission or immune rejection, whereas in case of whereas the disadvantages, there is only limited availability, and you would be injuring the site from where you are harvesting the cells. Allogenic cells have greater availability and are less expensive. However, there is risk of immune rejection and disease transmission, and you would also end up with a heterologous population when you use cells from another individual. Xenogenic cells are cells from other species, which means they are the most abundant and the least expensive. You can actually get it from organisms which can be grown for harvesting these cells. But there is a much higher risk of disease transmission and immune rejection. It is important to balance these advantages and disadvantages. People do try to use autologous cells as much as possible. However, it is not readily available. In many cases, what people do is they try to harvest autologous cells and then culture them, expand them to get desired numbers, and then use it for treatment of the injured site. In the introduction lectures, we had actually talked about using autologous cells for autologous chondrocytes for regeneration of cartilage, right. Those kinds of things are very commonly done even today. Differentiated cells and stem cells also have their own advantages and disadvantages. Differentiated cells have the required functionalities which are needed for the tissue to function the way we expect them to. Disadvantages, you would cause donor site morbidity, and in vitro growth is actually more difficult. It does not you cannot culture them for multiple passages, they actually die after some time. They are more expensive. Stem cells on the other hand are less expensive, and there are different sources from which you can get stem cells. They can be used for many applications. If you have one type of stem cell, then you can try to differentiate it into different cells and use it for different applications. Those are some of the advantages of stem cells. However, the disadvantages, they do not have the functionality, which means you need to differentiate them properly to get the desired functionality, and they may not do that, they may not differentiate as required. There could always be an issue with differentiation, especially when you have when you expect differentiation to happen in vivo. So, in vitro you have a lot more control with what the cells are exposed to and how you actually treat the cells to get the required differentiation, whereas in vivo the level of control you have is lesser. Okay. There is also a risk of uncontrolled growth and differentiation. So, this is especially true for embryonic stem cells, which might lead to formation of teratomas, which could be a problem. So, the limited availability of differentiated autologous cells is one of the limitations why people have started people looked for stem cells and other sources, and morbidity and limited proliferative capacity are serious limitations when you are talking about expanding the cells. As I was saying, we would not be able to harvest enough cells, right. So, if you are trying to harvest a lot of cells, then you will cause significant damage to the donor tissue. So, you try to harvest only smaller numbers and then you have to expand them, and expanding them is also a problem because they do not grow as rapidly and they only multiply for a few passages, they do not multiply for a long time. So, that is a problem. So, because of that using autologous differentiated cells is not very easy for many applications. So, with respect to undifferentiated cells, the limitations are different for the different kind of stem cells you use. With embryonic stem cells, there is a chance of teratoma formation. Teratoma is a tumor tumor containing tissues which is derived from 3 embryonic layers, which is the endoderm, mesoderm and ectoderm, and there is also a chance of uncontrolled differentiation in vivo, and these are technical difficulties, and you also have ethical questions when you are talking about embryonic stem cells, right. Mesenchymal stem cells have a problem because they fail to differentiate into desired cell types in vivo, even though people have repeatedly shown that in vitro differentiation is very successful. So, because of this, it is not very easy to use these stem cells for in vivo applications. So, many a times what people do is they use stem cells and differentiate them, so they expand them and then differentiate them in vitro and finally then place them in the site of implantation. So, those are some of the challenges with stem cells. So, what are stem cells? So, stem cells are cells that have the ability to divide for indefinite periods of time in culture and can give rise to specialized cells. So, they themselves do not have any special functions. So, they are undifferentiated and they can differentiate into different types of cells. So, there are 3 different types of stem cells. They are called as the totipotent, pluripotent and multipotent stem cells. So, totipotent stem cells are the cells which can form all types of cells in the body and the extra embryonic cells, so which are the placenta, right. So, it can form all of that, and embryo and the first few divisions of the embryo are the totipotent stem cells which can actually form any type of tissue. So, the totipotent stem cells have the ability to actually develop into a whole organism, whereas the pluripotent stem cells can form all the cell types in the body, but they cannot actually form the extra embryonic or the placental cells. So, those are the pluripotent stem cells. So, embryonic stem cells and induced pluripotent stem cells are examples for such pluripotent stem cells. Multipotent stem cells are the ones which can develop to more than one cell type, but are more limited than pluripotent stem cells. They cannot form all the cell types, but they can form a significant number of cell types. Okay. Yeah.
So, are stem cells in umbilical cord multipotent?
Yes. So, the cord blood cells are multipotent stem cells, only embryonic stem cells are pluripotent. So, totipotent cells can differentiate into extra embryonic membranes and tissues and the embryo and all the post embryonic tissue and organs, which means it basically has all the potential to develop into a whole organism. So, embryo is basically a totipotent cell. So, the stem cells are broadly classified as adult stem cells and embryonic stem cells based on where you get it from, right. So, if you are getting it from an adult organism, then it is an adult stem cell, and if you are developing an embryo from which you are harvesting these stem cells, then it is called an embryonic stem cell. Adult stem cells are actually present in different parts of the body. So, they are multipotent, and these were discovered almost 50 years ago and more than 50 years ago now. Embryonic stem cells are found in developing embryos and fetuses. These are pluripotent, and these were first isolated in 1998. They can multiply more readily and seem far more proficient compared to adult stem cells. So, when you are talking about the three major types of stem cells, the totipotent, pluripotent and multipotent. So, the potency of the totipotent stem cells is much higher than that of pluripotent and multipotent, and cell types which are capable which they are capable of generating, totipotent can generate any type of cell including the extra embryonic cells, whereas pluripotent can differentiate to form all the three germ layers which we talked about. So, whereas it cannot form the extra embryonic tissue, and multipotent is limited to certain cell types. So, examples are zygotes and early morula, which is the first few cell divisions of the embryo, and embryonic stem cells would be pluripotent stem cells, and induced pluripotent stem cells are also pluripotent. Hematopoietic cells, mesenchymal stem cells, neural stem cells are all examples of multipotent stem cells. So, these are basically different types of adult stem cells, and totipotent stem cells are found in the fertilized egg and the early cells of the fertilized egg, and pluripotent stem cells, which is the embryonic stem cell, is found in the inner mass of a blastocyst. An embryo develops for a few days and it forms something called a blastocyst, which is a cell mass. The inner cell mass of this blastocyst is embryonic stem cell, and multipotent stem cells are obtained from different tissues. The advantage of each of them is totipotent stem cells are easy to isolate and grow, and pluripotent are also easy to isolate and grow. So, you can get a lot of them, it can easily be cultured. But with multipotent stem cells there is less ethical issues, and there is less chance of immune rejection because there is a potential for it to be taken from the same person, right. So, you can get an autologous stem cell and actually culture it and use it. So, cord blood cells and things like that, that is why people store cord blood, hoping that they would be able to use these multipotent stem cells eventually. So, the disadvantage is with totipotent and pluripotent is the ethical issue, and with pluripotent stem cells there is also chance of teratoma formation, and multipotent stem cells are very difficult to isolate, and they have limited differentiation, and they are not available in large numbers. So, when you aspirate blood from bone marrow, it is not very easy to get stem cells, you get only a very few stem cells for the aspiration you get. So, those are some of the problems. So, teratoma is like a cancer tissue which contains tissues from all the three derm layers. Okay. So, it is a problem with embryonic stem cells specifically, and iPSCs may also have the same problem, but people are trying to understand that. So, see, adult stem cells are primarily obtained, large source is the umbilical cord blood or tissue, and there are also cells from brain, cornea, retina, heart, fat, skin, dental pulp, bone marrow, blood vessels, skeletal muscles, intestines, everywhere, right. So, these are just places where you get different adult stem cells, and the number of cells you harvest will depend on the source itself, right. So, umbilical cord obviously has the most abundant, and bone marrow has significant numbers, whereas if you were to take it from dental pulp or other places, the numbers are usually smaller.
Sir, I am not sure, I read somewhere that recently they isolated stem cells from teeth.
Yeah, that is the dental pulp.
Oh.
So, the teeth has a pulp in the root, and that is where you can actually harvest it from. So, embryonic stem cells are obtained from fertilized egg, usually through in vitro fertilization. The ovum has had a nucleus removed, and a nuclear membrane material is injected from the intended recipient, which is called, it can be reproductive or therapeutic cloning, to get the in vitro fertilized eggs, which you culture for a few days and then finally get it. So, the question is always about the ethics of this. So, some of the unique properties of stem cells is they are capable of dividing and renewing themselves for long periods. So, stem cells can multiply for many months in the lab, and they are capable of long term self renewal, in the sense that even when they are differentiating, they will maintain the numbers. and they are unspecialized, which means they do not have any tissue specific structures. So, they can actually be differentiated into different types of cells. They can give rise to specialized cell types which will happen through the process of differentiation. So, differentiation actually does not happen as one step, it goes through multiple steps where the cells become more and more specialized, and finally, they get committed to one lineage and form one particular type of cells. So, adult stem cells typically generate the cell type of the tissue where they are obtained from. So, if you take hematopoietic stem cells, it will form different types of blood cells, right. So, it can be differentiated into other cell types by forceful differentiation, but in general it can differentiate only to those types of cells. Okay. Adult stem cells are undifferentiated cells which reside in the differentiated tissues. Progenitors and precursors are formed before you fully form the fully differentiated cells. These are the intermediary steps of the differentiation process. These are progenitors are however committed to a lineage, whereas the stem cells are not. So, that is the difference. So, although in some cases people do try to use them interchangeably, it is not scientifically correct. Progenitor is already committed. So, one example would be endothelial progenitor cells. So, these can only form endothelial cells, right, but they are not fully developed into endothelial cells, but they can actually grow at a much faster rate compared to endothelial cells. So, the function of adult stem cell is to maintain tissue homeostasis and replace the cells which are lost due to normal tissue turnover, injury or disease. Bone marrow is where the first set of stem cells were identified, and hematopoietic stem cells were identified in 1961 and 63, and these can form the RBCs, WBCs and platelets. And bone marrow stromal cells were identified in 1970, and they have been shown to form bone, cartilage, fat and hematopoiesis supporting stroma and so on. So, stem cells have the self renewal property because of something called asymmetric division. So, what happens is whenever you have asymmetric division, there are two daughter cells which can have two different cellular fates. When you have mitosis, the two daughter cells formed are exactly identical and they have the same fate. Even with meiosis, that is what happens, right. Meiosis also, you form two daughter cells which have the same fate, they can only be gametes, they cannot be anything else. Whereas here that is not what happens. You have asymmetric division where the stem cells which divide, one will be a stem cell, which means the self renewing property is maintained. So, one stem cell forms one stem cell and another cell which can get differentiated into any type of cell. So, your stem cell population remains the same, okay. The number of stem cells in their population remains the same. So, basically one daughter cell has the same potency which is the self renewing property. So, it is a stem cell, and the other one can either be a stem cell or be stimulated for further differentiation. So, usually what happens is this daughter cell which has a different fate can actually multiply before it gets differentiated. That is what happens, because after complete differentiation, if the multiplication has to happen, then it will be much slower. So, multiplication usually happens before complete differentiation. So, that is why these progenitor cells are important. So, these progenitor cells will actually divide at a faster rate, but they are already committed to the lineage. So, they will not come back to the previous step, they will actually get differentiated to the final type of cells. So, hematopoietic stem cells are the ones which are obtained from the bone marrow. So, the way you harvest them is an invasive procedure. Obviously, you have to drill into the bone marrow to get it. So, it is not a very preferred mechanism for harvesting, but that is the only way you can harvest hematopoietic stem cells. They can also be released into peripheral blood by a process called mobilization. So, this mobilization can actually be induced by treatment with cytokines, and thereby you can actually get them into the peripheral blood and then try to draw them. So, blood from the placenta or umbilical cord would also have hematopoietic stem cells. These are much easier sources to harvest these stem cells. Mesenchymal stem cells are the ones which are extensively studied. They are obtained from the bone marrow of the iliac crest or the femoral head from a patient who is usually undergoing total hip replacement. So, you do not harvest mesenchymal stem cells only for that purpose, because it will be a quite a painful procedure. So, iliac crest is from your hip, and you can get it from your sternum, which is the joint between your rib cage, and you can also get it from the femoral head. Femur is the bone in your thigh, and the head which gets into the ball and socket joint, that is also that is the head from where you can get these. And adipose derived stem cells can be obtained from fat tissues, usually which is taken after liposuction, and adipose derived stem cells have also been differentiated to various types of cells, and they have been effective in using for tissue engineering applications. Epithelial cells are usually found in the bulge containing region of the hair follicle, and these, as I said, the numbers would be small, it is difficult to harvest them, but they do have the same multipotency as mesenchymal stem cells. Other stem cells are neuronal stem cells, multipotent adult progenitor cells, and cells which are present in your connective tissues, and muscle derived stem cells. So, these are all the different adult stem cells which have been used for different tissue engineering applications. So, multilineage potential of the adult human mesenchymal stem cells was demonstrated in 1999 by Pittinger et al., and this was published in Science, and this is one of the extensively cited papers, which has almost 23,000 citations currently. And so, these are cells which were previously called as a plastic adherent colony forming units, fibroblasts. So, the term mesenchymal was coined in this 1999 paper. So, what happened was Pittinger et al. Demonstrated that these human mesenchymal stem cells have a multilineage potential, and they form adipocytes, chondrocytes and osteoblasts. So, they also showed that these mesenchymal stem cells had heterogeneity with growth rate, phenotypic plasticity and colony morphology when cultured in different conditions. So, initially they were identical, right. So, when you harvest these mesenchymal stem cells, they are all identical, all the morphology, all the properties are the same, but when you culture them in different conditions, they became different cells, completely different, and that was shown to actually that was shown for the first time in this paper. So, stem cells are present in your body in a place called stem cell niche. So, this is a microenvironment which is well controlled to maintain the stemness of the cells. Okay. So, this is a specific location in which the cells can reside for an indefinite period of time without actually differentiating. So, they can produce progeny while self renewing, in the sense that they can go through asymmetric division, and the cell which is fated towards differentiation will leave the stem cell niche, and the cell which is going to remain with the same multipotency will remain in the niche itself. So, this is one of the issues when we handle stem cells. We do not have the stem cell niche outside the body. So, you can have the stem cells, but they do not behave the same way. When you have it in a stem cell niche, they have certain properties which are which will be lost when you take it out of them. So, there are different types of stem cell niches. So, you have a simple stem cell niche. So, these are basically niches where the stem cells are locked to the niche by adherent junctions and to the extracellular matrix through different junctions like integrins. So, the niche position of the stem cell is to receive intercellular signals that control the growth and inhibit differentiation. So, in a complex niche, different stem cells might be localized in the same niche, and more cell types can contribute to the niche instead of just one single cell type contributing to the niche. The storage niche is the stem cell niche where the cells are just stored, the cells are quiescent inside this. These act as the reserve of stem cells and they are activated only at, during injury or some kind of damage to the tissues, so that these cells can actually go and repair the injured tissue. So, progenitor cells are cells which are more specific than the stem cells, and they can differentiate for a limited number of times. These are not self renewing, but in some literature you would see that they are using it interchangeably, which is not correct. Endothelial progenitor cells or pancreatic progenitor cells are cells which are committed towards the particular lineage. So, embryonic stem cells were first isolated from mice in 1981. So, it was done by two different groups independently, and in 1998 human embryonic stem cells were isolated by Thompson et al. And these are isolated from the inner cell mass of the blastocyst and can be expanded in the lab while maintaining the pluripotency. So, this is what is done. So, for isolation of embryonic stem cells. So, the you see, to get the embryo out of a, so if it is natural fertilization, you would have to get the embryo out of the ovarian duct, right. So, then that would mean flushing of the duct, which will destroy the embryo as well. So, that is not really an option. So, because of this you need to do in vitro fertilization. That is how you create these embryos for getting these embryonic stem cells. So, surplus embryos during IVF treatment can be used for this. So, this needs to be donated by the parents after informed consent. So, so in India you cannot pay for these things as well. So, whereas abroad people do pay for some of these donations which people make. So, even when for blood collection in the US, we used to go and give blood very commonly for research, because many of my friends were working with blood and they would need 30 ml blood for research, and so they will call one of us and we will go and give blood. We will get like $20 or $30. So, that we used to do that quite regularly, but in India you are not allowed to do that, because you can exploit the poor in name of doing that. So, in the US you only exploit the grad students. So, so zygotes are produced through in vitro fertilization, and they are cultured to form the blastocyst phase, and instead of, so at this stage usually what happens is this is placed inside the womb for it to develop into a fetus. So, here instead of that the blastocyst is used for isolation of embryonic stem cells. So, there are only about 200 to 250 cells in the blastocyst, and 30 to 35 of them, 34 of them is the inner cell mass, and the outer layer is removed, and either through mechanical surgery or through immunosurgeries, and after that you take the inner cell mass and culture it on a feeder layer, and the colonies are mechanically dissected and transferred to new dishes. So, these need to be cared for daily, so that you make sure that the differentiation does not happen, because when you are culturing it in vitro there could be stresses which are caused that could cause differentiation. So, to maintain its stemness you need to make sure it is cared for and it is maintained in the way it can be. So, you would always prefer to use, this is for any cell. Yeah.
What was that feeder layer?
So, feeder layer is basically another cell, another monolayer of cells which is used, on which the cells are cultured. So, usually they are fibroblasts. Fibroblasts are cultured as feeder layers. So, this goes for any cell type. Fresh non-frozen cells are always preferred when you are talking about primary cells compared to frozen and thawed cells. So, the cell lines which you use are different, those are immortalized. So, those, yeah, you can put it in liquid nitrogen and then take it out and use it, but for primary cells you would not want to do that. So, that is why you need to harvest them regularly. So, this means it is a problem, right. It is you are not going to get enough cells with respect to embryonic stem cells. So, there are also issues with respect to long term stability, and there are cases where there is early differentiation. So, when you culture these cells in 2D environment, there is, the cells can actually pile up, right. So, instead of forming a monolayer, once there is more cells it can start piling up, and this will create stresses causing differentiation around the edges or in the piled up areas. So, these places would experience more stresses and it results in differentiation of cells. So, you can actually use endogenous transcription factors to activate the cells differentiation, and this using different growth factors, using different cytokines and other markers, you can actually control how the cells are differentiated. And you can also co-culture with cell types which are capable of lineage induction. So, there are studies where people have shown that culturing endothelial stem cells along with pancreatic cells, islet cells would cause them to differentiate to form islet like cells. So, those kinds of things are possible. So, iPSCs are the last type of stem cells which we will quickly go into. So, these are basically somatic cells which are differentiated into induced pluripotent stem cells. So, this was first shown in 2006 by Takahashi and Yamanaka, and what they showed was there were a set of 4 genes which can actually be transfected into the cells to form them into pluripotent stem cells. So, human iPSCs were then reported a year later. So, these cells have very similar properties as embryonic stem cells. So, Yamanaka won the Nobel Prize in 2012 for physiology and medicine for this discovery. So, mouse iPSCs demonstrate certain important characteristics which are seen in pluripotent stem cells, basically expression of stem cell markers, ability for formation of teratomas which are tumors containing all 3 germ layers, and the ability to contribute to many different tissues when injected into the embryo for at early stages of development. So, human iPSCs also have the similar markers and ability to form cells from all 3 germ layers. So, there is a lot of studies going on in this direction, and it is a very recent development, right. So, less than 12 years old. So, it is a very interesting domain.